Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. And once again, please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It is my honor and privilege to welcome our speaker today, Dr. Randy Golub from the McCann's Center for Brain Health at Massachusetts General Hospital in Boston, Massachusetts. Dr. Golub earned her PhD and her MD from Duke University School of Medicine. She conducted a residency at the Hospital of San Rafael in New Haven, Connecticut, and another residency at Yale New Haven Hospital. And she has board certifications in psychiatry, as well as through the American Board of Psychiatry and Neurology. Dr. Golub was recently elected to the chair of the Organization for Human Brain Mapping and serves as professor of psychiatry and associate director of translational research in Neuroimaging research program at Massachusetts General Hospital. She is a recognized leader in the development and applications of advanced neuroimaging technologies to understand the pathophysiology of neuropsychiatric disorders, including neonatal hypoxic ischemic encephalopathy, chronic pain, and schizophrenia. She is actively working on translating these advances into clinical radiological practice to improve patient care through the use of large-scale imaging informatics approaches. Work in her laboratory focuses on using clinically acquired brain images from neonatal subjects as well as young children to build age-specific normative brain atlases of quantitative tissue metrics to advance understanding of brain development and the application of those normative atlases to detect abnormalities associated with hypoxic ischemic encephalopathy in neonates. This approach enables the detection of subtle anatomical variations through the use of machine learning algorithms and other sophisticated statistical techniques. The work of Dr. Golub and her team demonstrates the exciting potential of leveraging existing electronic health records data, including clinically acquired brain imaging scans, to yield information on par with clinical research studies that were conducted over decades at the cost of many $million. Dr. Golub's lecture today is entitled Clinical Imaging Informatics in Action, Extracting Valuable Knowledge from Archival Pediatric Brain MRI. And as always, we are streaming this lecture live and for recording via YouTube. If you are joining us via YouTube, thank you so much for your attention today. Also, our specifically selected 2020-2021 Biomedical Data Science Innovation Lab participants are encouraged to submit any questions for Dr. Golub via the chat feature in their Zoom sessions. I will synthesize these questions and I will ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Randy. We are so excited to hear your lecture today.
Well, thank you, Jack, for that lovely and friendly introduction, and good afternoon, everyone, or whatever time you're watching this. It is my pleasure to be here and to share this fairly unique kind of research with you all. I know for those of you who are in this program, some of you are trained as clinicians and certain features of this will be a little bit more familiar and others not. So I'll hopefully take the time to help everyone be on the same playing field to understand how this is done and why. In particular, I'm going to try and make the case in a way that will be completely understandable as to why someone would want to bother to use the clinical imaging data that's collected for secondary research purposes. I'm going to help you to understand what the challenges are and also show you some exciting new developments that are beginning to overcome some of those challenges. I'm going to show you at our institution how exactly we can make this happen. And then by way of sharing the exemplar work that's being done with my colleagues, help you to have a real intuitive understanding of what could be done and its potential, with just a glimpse at what the future might hold. I just want to take this moment to give a shout out to my friend and colleague, Steve Piper, who, if you notice in this little front picture here, each of these, this sweet baby's face is made up of a mosaic of these little thumbnails of brain image data. This is sort of a very dramatic illustration of how much of those image data we have available to us to use to do this research. And Steve is a master at these things. So the why part is really important. Why bother? You've heard recent talks. I believe Jessica Turner spoke with you not too long ago about the Enigma, all these different huge efforts that are being underway, that are being funded to collect lots and lots of data for the research community to use. Well, there's other data also, and there are special reasons why this data might be more appropriate for something that you're interested in doing. So for example, if you quickly need to get a hold of, say 200,000 brain MRI scans that you wanted to be able to extract a hippocampal volume from in order to build an AI tool that would help you to screen for early evidence of cognitive decline. Well, this path that I'm going to show you might be one, it's not free and it's not immediate, but there is a path to get that data to work on. And in particular, because this is real world collected in the wild data, the tool that you would develop using these scans might be much more immediately useful to run on the standard clinical scanners that are in use in the different academic healthcare centers or hospitals, worldwide. And as opposed to tools, many of which get developed on the research data, and those early passes really have limitations to how well they will operate on this tougher data, I should say. I'll be showing you some things about that today. Another really important thing to consider is what kind of data from which patient cohort or what kind of ages are necessary for the work you want to do. And in some cases, if it's a rare disease, it may be very hard over the time scale that you might have available on a grant or in your lifespan to collect everything yourself. So there's collaborations, but there's also ways of acquiring that data through the hospital archival scans. In the case of what I'll show you today, it's that rare data from being able to get a hold of information from brand new babies who typically are not in a hospital having their brain scanned. And while I'll talk today about all brain MRI, because that's the domain that I work in fact, this workflow, this sort of conceptual frame for what I'm showing you would be completely appropriate if you needed a CT scan, if you needed an ultrasound, if you, whatever kind of imaging that you needed to use. The workflow that this data comes in under is an important concept to understand in terms of, you can think of it as your experiment of how you're enrolling your patients. You'll see how this matters. In the typical clinical workflow, a scan would be collected because some doctor has a patient that they feel needs to have some imaging done to help them care for their patient. They enter an order. A unique accession number is assigned to that to identify the study in the patient. It's scheduled. This protocol step is really important. That's where the radiology department or the other domain expert will choose explicitly how the scan will be collected. Always it's done with respect to what is the question being asked of that patient. When the patient arrives, they are brought to the scanner. That information is used to label the scan. And during the scan session, if the operator notices something that they weren't expecting, they might add or change that protocol by adding additional scans, or if there's a problem in the system, they might repeat a scan. And then those images are passed to this archival storage, the Picture Archive and Communication System, the PACS, which is a very smart database because it supports a lot of communications. All the images are in a file format called DICOM. Those of you who work with any kind of medical images are probably familiar with this standard. It's whole lectures on these topics right here. And depending on the study, they may already be automatically based on the order that was entered and the exam that was done, sent for some additional advanced image processing to feed that information also back into the PACS. And then it's fed into the workflow of the radiologists who will read that information, read the scans, interpret them, and fill out a report that is then also put into the medical record. And so people working in EHR data have access to many of these different bits. They accept these images. Typically, when a clinical informatics person is working with medical imaging data, what they're really working with is doing some NLP on the exam, on that interpretation that the radiologist has provided, and not digging very deeply into the medical image data itself. So I wanted to share a force vector that suggests that this work could be very meaningful and continue to be more impactful over time as the fields of research imaging and clinical imaging. They're always interacting with each other as the quality of the scans improves through higher temporal resolution and spatial resolution and different types of sequences that give more and more information that's meaningful about tissue properties. The size, the magnitude, the amount of data that's embedded in a scan continues to grow. And so the requirement to extract as much information out of that also continues to grow. And so this move towards more quantitative assessments with tools that mine the data and then use the information as it's coming in to help create more useful information for how to care for a patient builds on this informatics infrastructure. And so the whole field is definitely moving stronger and in many supported ways towards this more quantitative extraction of meaningful data out of the medical images, which supports the effort that we're doing because it makes it easier to find and work with and do more with that data and makes the results that we create on the research side gives us a tighter and stronger loop back up into the clinical care. So just a quick digression just to say that these standardized radiology reports, this templated reporting is also something that is of value in that it helps that when a AI tool quantifying hippocampal volume happens, that the radiologist has a way to easily and quickly insert that information into the medical record, making the EHR data more and more rich and easier to use for research, but also enhances the quality of what is available for the clinicians in those radiology reports when they're making their decisions for patient care. And so the incorporation of that additional information is eased through these structured reporting. So all these different efforts on these different fronts kind of are all sort of lifting us up and making it more and more likely and more reasonable that this work will have a clinical impact. And in the end, that's what matters, so much so. So now, coming back from our digression, so the unique challenges if you want to use this kind of data for your research. If we look at the parallels in this domain, you might be used to in the research world, thinking about designing a study where you would have a standardized acquisition protocol. You'd be very specific about it, and all the little details of how it would be, how a brain MRI scan might be collected would be decided in advance. You'd have maybe make efforts to send a human phantom to the different sites to make sure that they were all calibrated as perfectly as you could. You go for the best possible image resolution. You carefully screen your subjects. If you have a patient cohort, you use detailed inclusion/exclusion criteria to select them. You carefully match the healthy subjects on all the different demographics that would need to give you the best bang for the buck and carefully comparing your patients to controls. The results you get very quantitative and standardized. So when we go through on the clinical side, we approach it very differently because, as I just taught you, for every time an acquisition happens, it's really patient-centric. It's based on the needs of the patient and customized to them. And so, while there is some systematic effort in there because there are just a sort of a finite number of protocols and sequences that could be run, the recipe is, absolutely tweaked per person. And an important issue here is time versus money. So the more resolution, the higher the resolution, the more detailed, the more complete the anatomical coverage, all of those things map to taking longer time, not only to acquire, but for the radiologist to read and interpret. And so the data sets, as they're acquired clinically, are matched to the clinical question. If a person is being ruled out for a stroke, they're going to get a very fast, very rough scan because strokes show up really well on these scans. And so the minutes that you might take to get a better answer will be potentially lost brain or more brain damage, when the real need is to get the person quickly into the suite where better imaging is available, but they'll have it as they are already doing an intervention to save the quality, to save that person's brain by opening a clot or stopping a hemorrhage or whatever they're doing. And so the variability in the quality of the scans is quite high depending on the question being asked. If you're going in for a pre-surgical mapping for an epilepsy problem, you might have super high resolution because they're looking for these tiny little cortical dysplasias, for example. So on the subject level, very rarely is any data normal because no one is getting scanned because they're normals. They're not, they're getting scanned because they're trying to rule out a problem. But sometimes there aren't problems, and it's very reassuring to the patient and the clinician that they didn't find the problem and they look for something else in a different way. And so there are a few scans in the packs that are healthy and don't have brain pathology. And what you get out are those radiology reports, which are just a mere fragment of fraction of what's available in the data. So here's a few just representative, images randomly pulled out from our study, just to show you that the quality of the data that's in there is really quite nice, and there's lots of it and all different kinds of it, and it's awesome. But not all of it. So more often than in a research study, there will be poor quality because of undersampling or motion or some kind of a noise artifact. And I'm going to do it again on one of my little digressions here to just show you right now, the way that we deal with this is we just have to exclude these studies because we can't use them. But there are some really exciting novel approaches that are on the horizon to mitigate this damage or correct them afterwards, and work that's being done partly by some of the students and faculty where I work and many other places, I think probably around the world. But I'm going to teach you one tiny bit about MR physics, which is just to say that that this work that's being done by our student Nalini Singh, it's a really cool work, relies on the fact that at the time that a patient is in the scanner, when the scan data is being collected, it's collected in what's called frequency space or K-space. And a Fourier transform is done to take the data out of K-space and generate the image. In a typical MR workflow, this happens with one pass, it's done, and then that's what gets loaded into the radiology archives. So here is an experiment that she has been working on where she's actually using some machine learning tools to use both the K-space and the image space data together using a deep learning joint algorithm that takes those two inputs, the K-space image and the K-space and the reconstructed image, and uses them to iteratively improve the quality of the final image that would be put into the radiology archives, for example. So what I'm showing you here in the data set she uses for this research, she has what is called ground truth, which is a very high quality, beautiful scan. Here, down here at the bottom row, you see the frequency spectrum. That's the K-space that would have come off the scanner. In the little red box is a higher resolution, not a higher, but focus in on this red box is what's shown in this little patch right here. So you can see more of the anatomical detail. And when she takes the data set from the same subject that's been corrupted by this motion, the person was moving their head up and down, you can see that in the, especially easy to see in this higher field image, that there is a ton of loss of information. This difference line here shows you the difference between what's in the high resolution, the ground truth, and this input image. Here's what the K-space looks like in that corrupted image. So after it goes through her magic, I'm not even showing you the black box, this empty white space is her algorithm. What it spits out is an image that looks very close to the ground truth. You can see it higher resolution here, and in the difference image, you can see it's not perfect, it's not exact, but it is considerably better than what you see in the original image. And so this is future state because this would have to be done at the scanner when you still have the K-space available, the K-space data available in a standard workflow. Once the Fourier transform happens, images are saved, but the K-space data is thrown away. And so you can't work backwards. It's super cool work, and there's a citation if you want to learn more about it. Similarly, some of my other colleagues, this is work led by Eugenio Iglesias. He takes a post-acquisition approach. And in this case, it has the possibility of actually rescuing scans that were collected, enabling us to do more quantitative morphometry on that data, or morphometric analyses on that data. And this is maybe a little closer on the horizon because we can use it on existing scans. So again, this is work that requires the training of these deep convolutional neural nets. where inputs, they train a model using good quality data and generate models, but then taking these poorer resolution or noisier scans, as I'm showing you here as the inputs into that trained algorithm, can output something that is a little bit closer, actually considerably closer to what ground truth would be. This work is being done on a data set that is the ADNI data set. So he has both ground truth and the degraded images to work with. To make this a little more concrete, here's what a scan, this is an axial scan through the brain, and it's showing you the output of the software tool called FreeSurfer, which is able to both segment cortical and subcortical structures, but also make a very high resolution assessment of the cortical surface and the peel surface and generate these nice surface maps where you can see cortical thickness and curvatures and things like that. In data that's collected, say a more typical clinical acquisition would be thicker slices. So this might be 7 millimeters instead of 1 millimeter in thickness. Here, the algorithm doesn't do, the FreeSurfer tool does not work so well. But after he runs his convolutional neural network on that data and spits it out, it does bring it right back much closer to what the original, the known ground truth in this case, this research data set shows. And so here quantitatively, with respect to that hippocampal volume I was speaking of before, when you look at how accurate the reconstructed data is, or a lower resolution data is, and how accurately it can find hippocampal volumes, what he has been able to show is that with his nice algorithm, the amount of error goes from, in a 7 millimeter scan, goes from the scattergram of these red dots to this scattergram with the blue dots, making the average error dropping from 10% to 3% and increasing the effect size. Basically, what that means is you have a much greater sensitivity to detect an abnormality that is due to the true anatomy, anatomical problem. And so, while this has been done on one test data set, this is again also a recent report, so early days, lots of exciting to look forward with that. Okay, back from those digressions about cool tools that are going to help us in the future. Let me show you how we do this at my institution. And so we have so I don't know how many of you at your centers have a clinical informatics infrastructure. Maybe you have the one called I2B2 or some other ones where you're able to do a lot of research using the nicely archived clinical data. Where I work, this is called Partners. Now we call it MGB for Mass General Brigham. We have a really robust set of tools where we can actually find patient cohorts, and work that I've done collaboratively with my colleagues in radiology and this group's team, as well as Dan Marcus at WashU. We designed and built a module, a plugin to it that allows us to actually pull those medical image data out. So reminding you of the slide I showed you a little earlier, the RPDR tool is allows us to pull out these demographics about the patient and their accession numbers and the radiology report and the medical record numbers and all that stuff. Whereas the B2B2 tool allows us to actually go in and query and retrieve from the PACS the actual image data. So how does it work? You are credentialed in. Here's my colleague, Sean, who's my co developer in arms for developing this capability. He is the visionary behind this whole RPDR infrastructure here. But this is basically a query front end for a big database, a huge database with something like 6 or 7 million patients worth of records in it. And I don't know how many hundreds of millions of pieces of information it sits in front of, but you basically just click on little folders that represent the information you're interested in to drag over and submit a query that is the equivalent to you designing your cohort experiment. So you can choose a cohort based on a diagnosis, a set of diagnostic codes, or on a set of procedure codes, or a set of lab values. You can include or exclude them. You can restrict by age groups. You can do all sorts of very, very sophisticated analyses to generate a query. And you don't even need an IRB at this point. What you're doing is just trying to understand what the sample size is that would meet your criteria, and kind of does it break out demographically to be what you want. Because once you generate the query that represents the cohort you're interested in, you click this request detailed data button. And actually that was an older slide, an older version. Now we actually, this is the same slide that I had just showed you, but now in our newer, more enhanced version, in addition to requesting the detailed data, which would just be what I told you RPDR spits out, we have another button that says request those. This is how you get your Me2B2 workbench. That stands for Medical Imaging Informatics Bench to Bedside. Me too, I want my images, is how I named that thing. So this is where you click this button in order to put in your request for the brain imaging data. So this tool works really nicely because, if you are getting your images because of a query, it works just fine. But let's just say that you have an ongoing collaboration with a set of research colleagues who already know things about your study. So for example, in the work I'll be showing you shortly, some of my neuroradiology colleagues had been curating lists of healthy, normal babies and children because they needed them for teaching cases to teach the radiology residents. So when they would come across that rare once in a while person who actually had, or a child or baby who had a healthy, who turned out to be healthy and not have anything bad happen, like an example of a baby who had accidentally fallen in a swimming pool and was lifted out, and they were worried that they might have hurt themselves, might have been scanned, and then we had years of follow-up to say they were fine, for example. That would be the kind of thing that we might find. We had that list, we had a list of cases we got that way. So you can put that list in also, and then you put in a request for your Me2B2 workbench. And what's really important about how we built this is that access to both the RPDR data and the Me2B2 acquired images is managed in a way that's HIPAA compliant in order to keep a log at all times of under what IRB, by which investigators accessed which data sets, and they're all audit logged, and we keep track of all of that information to be able to report out. Once the person has their workbench, they log in, and through a series of steps, they sort of have to fine tune. Here's where searching in the radiology data, there's a little bit of work to be done there to find the actual sequences and the studies that you want, and you transfer those out for you to work with. There's citation right here, teaches you all about how we built this Me2B2 workflow, and actually has the link to, it's an open source code. So on the I2B2 wiki in the community pages, there's actually a project for us. And so any of your sites that have I2B2 that you would like to use this kind of software, you can. We are already updating it locally. So if you have any questions, reach out to me and I will happily talk to you more about it. If you're interested in knowing how to use this tool and what it takes and how to do it, there's really detailed tutorials on our help page, which is open. This is not behind our firewall. Anybody can see it. And it'll walk you through exactly how you use this tool to pull your data down. So that was the how you get to the data, but now let me show you some of the studies that we have done with some of this data that we began working on in about 2015, as we were pulling the data down, maybe even a little bit before that, and then even more recently. And this is really work that could only be done with the vision of my colleague, Ellen Grant, who sort of saw this whole arc of what I'm about to share with you in her mind's eye. And it's been a real pleasure being on this ride with her. And a lot of the work has been done by Yangming Wu. I'll show you more as we go. Okay, so for this, and this is work that you we'll be able to find that reference, but I just showed you how you can use the RPDR tool to query and find scans. And so we used a combination of the query tool as well as our data list, and we identified about 2,871 scans where we knew that they had had a brain MRI scan done. And we knew that they were between the ages of 0 and 6 years of age at the time the scan was done, the brain MRI was done. Now, one of the tricks here was that because Ellen is a neuroradiologist extraordinaire, she was the one who had actually set up the protocols for all baby brain MRI. And she had included a particular sequence. And we knew that one of the other criteria we used is the scan had been collected after, I think, 2006, because that's when she, very much before others were using diffusion weighted imaging, she was one of the pioneers of it. She had got the protocol set up and run and perfected so that it did the best possible job, and where a single protocol would actually work nicely across the ages of 0 to 6. And so over the many years since, the only very minor changes to that protocol happened, like when they would upgrade a scanner or the scanner software, but the basic procedure was continued all through those years. And so we had a very coherent data set to begin with. So we imported those scans from the PACS to our local workstation. And then, because we didn't have any tools that would help us get rid of the corruption that I was showing you, or other kinds of loss because of the data, we lost only, in this case, it's about not quite 50% of the data, which is an incredibly small loss. Most other studies that you might be going after to get good data, you'd have a much higher loss at this point. But we had a very good throughput because we knew how to ask the right question of the data that was available. So collaborating with your local neuroradiologist is really key. Okay, so then once we had gotten all of those scans, we had clinician scientists and nurse practitioners reviewing, and then our imaging scientists reviewing both the ADC scans themselves, those are quantitative apparent diffusion coefficient scans, and also looking into the EHR. We have, under our IRB, we had permission to not only download the data, but to go into the medical records to be able to see finer details, because RPDR doesn't have every detail of every patient in it. And some of the details you need to look in this pediatric data set, we had to look up on our own. And we used the REDCap database to collect all that kind of information in. And after carefully reviewing, we had to screen out. In this case, we're looking just for the healthy normative ones. We screened down to about 705 potential cases that might work well for us. And then, again, through this was just looking to make sure that the scans were normal enough. And then we had to do additional medical chart review on those cases to confirm that for several years afterwards, a minimum of 3 years, that the patients did not develop any neuropsychiatric abnormalities, to make sure that the image data that we were calling normative was indeed reasonably representative of normal brain functions and structure. And that got us down from 705 to 201. No, again, surprise that people who are imaged for a reason, for clinical reasons that are, where you are interested in finding out, or you have a clinical suspicion that there's something wrong with the brain. And even though at this point the radiology reports were read as normal or normative, that meant that the radiologists on their own could not detect abnormalities. It doesn't mean that they weren't there. It just means that this particular study did not reveal it. It was not sensitive to reveal it. And that's really, sort of a heads up, I'll come back to that, way that is telling you something. So if you're losing 500 scans because people do end up having abnormalities that you can tell already by the ages of 3 years old, then maybe there is some signal there that isn't being captured. And it's a commercial for what I'm going to tell you in a few more minutes. So getting down to the 210, we finally have nice clean data that we can use to generate some atlases, which I'm going to tell you about again right now, and use those in various ways to help the clinicians do better, make better use of this data. So the question is, how do you generate once we generate those atlases, how do we do good work with them and use them in ways that are really meaningful? And so when a research scientist on their own would go out and want to think about how do I generate these different brain atlases across these different ages, they might recruit a patient in and study them repeatedly over periods of time. So you could have longitudinal data, which gives you the most sensitivity to detect change over time. But then you're limited by your cohort size, and you can only generalize to people like the ones that you scanned. If you're interested in the span that goes from 0 years of age to 6 years of age, also the obstacle of scanning repeatedly in that age over that age span is very challenging. There are some good data sets out there and people working very nicely with them, but the approach that this data allows us to do is a very different one, where across time, we aggregate clusters of patients. Again, no two patients were used. Another reason that the numbers dropped, as I was showing you in the preceding slide, is sometimes there were repeat images on the same subject. So they might have been being studied longitudinally for clinical reasons, but we only used them once in our data so that we didn't have any biases. And so every single age, we have a cohort of patients that are the same age, and we can generate what an average, what a mean is of a different group each different time. The value of this is that, first of all, it's feasible, you can do it, and the costs of, you save a lot of money not having to recruit and scan, and you're also getting a better exemplar of what an average population might look like. So I'm going to spend a minute on this slide just to say this was something we actually were able to do in the early days, some of our very first efforts with this data. This is probably back in like 2014, maybe, where every one of these circles is a different scan result from a different individual baby or child. This is the data plotted from the first about 800 days of life, so very early on. And where there's colored markers and lines, those are cases where an individual actually had that longitudinal imaging going on. And a couple of things I want to illustrate here. So we had to be able to skull strip and somewhat normalize the data to be able to do this, and also strip away the CSF so that we could get what is called a whole brain ADC map. So this is averaging across all the voxels in the brain, the whole brain. And this WBA whole brain ADC measure. And what's plotted, what you see is how in the very earliest days, it's high and it starts to come down lower, which you can sort of appreciate here in that it's brighter and getting darker. One of the key things to notice is that this signal intensity is not, the ADC is not homogeneous over the whole brain. It does vary anatomically. And so the spatial localization of the value is really important. And what, when we were first gathering this, the first effort, Ellen had told me she was interested. She said, we should be able to find in this data a gradient from posterior to anterior, because this drop happens earlier in the back of the brain than the front of the brain. One of the features that makes this data so hard for clinicians to read and interpret is that these changes are happening so rapidly, especially in these early days of life. So interpreting what is normal, healthy changes due to aging and what changes are due to a pathological event is really hard to interpret. And that is part of the motivation for the work that we are doing. So I mentioned that I'd show you a little bit more about how those atlases are created. So taking those 201 total data sets, we divided them, binned them into age bins, the first 2 weeks of life, the second through fourth, second quarter, third quarter, fourth quarter, year 2, 3, 4, 5, 6. And in each of those cohorts, there were enough cases that we could, here are all the 13 cases, the 13 different babies that were used for the youngest age atlas. They were skull stripped and put into register and signal normalized. And then an atlas was generated that generated the mean ADC at every single voxel, as well as a set of standard deviation measures. And here are those atlases. This is the one for right here, the mean. This is just showing the mean of the ADC values over the brain in the coronal, sagittal, and axial views. And this is for each one of those different atlases from 1 to 2 weeks up through year 6. The way it's windowed here, it's a little hard to appreciate the subtle differences. But what I will tell you is that when we published this paper, it contributed tremendously to the literature because many people have been trying to gather this data, and there were bits and pieces in the literature reported here and there, but ours was the most comprehensive that covered this whole time frame. I'm going to do my normal, my well-deserved pitch to my colleagues who make this this resource available, that hosts the NITRIC website, which is a wonderful neuroimaging resource for finding all tools, all software, all data sets. Go there if you need anything to do with brain imaging. If you don't know about it, you should know about it. Everybody should know about it. But this is where Ying posted the software for how to make an atlas like this. And this is where those atlases are actually released in case you want them. And here's the most recent work. It took until just this past year to get published. First, this is hard, and second, neuroradiologists are really busy. But I want to call your attention to the fact that this was actually published in the journal Radiology. This is the journal that the radiologists use to help them advance their clinical practice. And by actually getting our study into that journal, which actually came with a really nice an editorial that said something about not for the faint of heart, because they appreciated how much work was done to be able to quantify this information. Like, not everybody can do this, be careful. But I want you guys to know that you can do this, and that this is a really valuable contribution to make. And so by getting it into this audience, we are able to show on both a sort of region by region way and also voxel by voxel. I'll show you in a minute how the brain matures over this age range, and to provide some quantitative metrics for what that looks like in these ADC maps that radiologists can use to help them benchmark their reads. And so here are those atlases that I had just shown you. And here are these beautiful segmentations, which Yang-Ming was able to make happen because he was able to use these atlas transforms to bring a segmentation atlas into this space and transform it into younger ages. Typically, these atlases require way more sort of detail in the primary image data to be able to find these anatomical features. And these ADC maps, they're very blurry. It's tough to see this kind of detail. And so having a tool that is meaningfully segmenting these multiple regions is incredibly useful, because it allows you to do something like this, which is to plot region by region how the ADC changes over age, where let me walk you through this set of histograms over here. What you see here is plotted, these are the ADC values, so you can, this is flipped axes from what I was showing you on the plot, my very first one, where I was showing you that little bit of longitudinal data in our cohort. But here is plotted the percentage of voxels in a particular ROI. The ROI here, the region of interest here, is the whole brain. So this is like that same whole brain histogram, and now it's broken out across the ages. So starting at the youngest, these are the highest values. So you can see this is sort of like watching the data come down in that histogram here in that plot that I showed you. Now you're seeing the histogram move towards these lower and lower values. But you're also seeing something really interesting. It's not completely smooth, and you're starting to see some little bumps here, which tell you that there may be some hint of that anatomical variability about when different structures mature. And so that's what's in these other plots here. So each of these numbers corresponds to one of these different plots, and each of those numbers is a different brain region. And but again, we're showing the changes in at each of the different ages over time. And you can appreciate those which show a slower rate of maturation along the scale compared to those that show a much steeper change. And so this kind of information is quantitatively useful to a radiologist as they are moving around in the space and trying to get a grip on what they should expect to see in their data. Here's a kind of a prettier way to look at this with 3D mapping of the same data, where what you're looking at is sort of the evolution of how this changes. And finally, with a lot of really elegant image processing capabilities, we can actually visualize that occipital to frontal wave of maturation that's seen in this ADC signal chain, ADC signal. So just really lovely. So let me just, I only have a little bit more time. Let me show you a clinical application of this work. When babies are born who have had basically a normal pregnancy, everything's great and fine, something happens right around the time of birth. And a full-term infant is born where there are many, this is kind of a grab bag term for what are many different reasons why a baby might not have enough oxygen or blood to their brain, which causes brain damage. It sadly occurs relatively often. And now we're starting to have much better treatments. For example, they figured out that they put the little baby in they cool the baby's temperature slightly, put them in these little cooling caps. They can slow down metabolism, they can slow down the damage that's occurring and help the baby to recover and save a lot of brain. But when this happens, it's not uncommon for there to be long-term outcome problems, neuro deficits of many different kinds. And so when the clinicians and the parents are trying to decide on the best possible care for a baby and for them, for the family, one of the tools that they use to understand what's going on is to get one of those brain MRIs, just like I showed you. And it's part of clinical, it's clinically required. And again, they are using those exact same acquisitions that I showed you, those really good quality acquisitions that have been stable. And not just at our site, but across the country and the world, it is standard that they use these brain MRIs to help predict long-term outcomes. And there is some amount of agreement, and it does help, and they do contribute and use the data, but there's a lot of variability, both within a single reader, like the same clinician reading the same study, you give it to them on other days and other times, they may not even get the same answer out, but very much between raters or different experts. And so even when people have a lot of experience doing this, it's hard for them to read these cases consistently. And so I've now taught you about those ADC maps, and in HIE, hypoxic ischemic encephalopathy, the damage shows up as lower ADC values, okay? And so sometimes it's really easy to see. And in this case, this is a healthy example, age matched to this clinical example. And probably all of you can make out easily those lesions that are not tough to read where there's damage. Most cases are far more challenging than that. And if I asked you to show me where on this scan or on this scan there were abnormalities, it's a much tougher job. And so how do we do that? We can take our atlases. And so here, is that ADC atlas from those little babies from 0 to 2 weeks of age. And here is one of those scans from a baby with a patient who has been diagnosed with HIE. When we at those voxels, and your eye can probably make out, I'm seeing a thing that says my internet's unstable. Hopefully you can still hear me okay. Maybe your eye can make out this darker area here. And maybe your eye can make out somewhat this little darker areas here and here. Maybe you weren't as able to appreciate some of these areas that were brighter than they might be expected to be. And so we're doing the work now to map how carefully and how accurately this algorithm by the algorithm being taking the atlas and comparing it to the patient's own scan, how often our atlases will detect the same thing that a radiologist here, 2 different radiologists manually marked up this patient's ADC map. And here you can see how closely the algorithm will compare to that. So this is one line of work that's going on right now. Another way that we can use this is to generate atlases from the scans of the babies who have the abnormalities and get some sense of information about which are the most commonly found areas that are abnormal, and or which regions or networks of regions are important for yielding the kinds of clinical pathology that we see. So finally, just a quick sort of vision of what a future positive might look like. If this work continues or things like it, then when the radiologist is in the reading room and they have a patient's data they're looking at, what they might also pull up in front of them would be a reference atlas that would tell them, so they could mouse over the 2 different ones and get a sense of what values should be there and what do I actually have here if I think I see something abnormal. And also potentially even a beautifully color-coded map that shows them where to double check more carefully. And it's still their choice what to make of this and whether that is information that's useful or not. And they have a lot more information they use. It's not to replace the radiologist. The name for this is computer-aided diagnosis. You give you use computational tools to help the clinicians to do their work. And so that's the kind of thing we're hoping to see, but maybe even before that it would be useful in classrooms for teaching people. So I will pause there for more questions. I will just give a huge shout out to Ellen and to Yang Ming, my friend Steve, the fabulous team that makes the our informatics tools work and sing at home. There's lots more people and good funding, and I'm really blessed to have had great trainees that worked with me from EPFL, and I miss them. I wish they were still back in my lab. It's a great team, and thank you for your time and for listening. Bye.
Randy, thank you so much for that. That was really exciting to see all that neuroimaging work. And some of those studies are, yeah, absolutely not for the faint of heart. That's a lot of work. I have a few questions for you. This goes back to the earlier part of your talk where you're talking about some of the image quality variation that we see in kind of clinical studies. And I'm curious, do you feel that there is a responsibility for like researchers to include or maybe even educate the MRI scanner techs as part of the research enterprise to make sure that they understand the value of getting relatively standardized data, even if they need to customize it a little bit for different sizes of head? You know what I mean?
So I would say that at most places, this is our teams here, at least at our hospitals, are so with the program. So we have a pretty virtuous cycle that the research teams that develop and enhance our new, because we are a big research site for Siemens and for other manufacturers, where we are always we are evolving and advancing the different kinds of technologies for later clinical, with a direct eye on clinical translation. So our teams of researcher, physicists, algorithm developers, whatever, are also very closely parked in our clinical scanners, helping to support the training and that transition and that translation. Our neuroradiologists are our partners in all of this, and everybody's pulling towards the best possible data in the best possible care of the patients. And so I think that there is I think that if you quietly took any one of our MR techs off in a corner and had a kind and supportive conversation with them, you would come away feeling like they really do know and they really do care and they really are doing their best.
That's super great. I think
it's really important to keep those conversations going. We work a lot on having like grand rounds and having case conferences and doing things like that. There's a lot of in-servicing. Communication is really important. And I'm sure there's outliers, but it's a team's, all of this is a team sport.
You shared with us some of the really interesting databasing developments that you've done there at MGH. And I'm very familiar with things that are large-scale neuroimaging data archives like the Human Connectome Project and various others of that spirit, where everybody, from the centers around the country are collecting all the same protocol and exactly the same kind of scanners and all that. So they're all very uniform. The clinical data, on the other hand, like for example, I have very often interested in traumatic brain injury data, but getting that data, there's very few places to go to find it. There are a few, but I'm curious if you have any other databases that you are aware of and want to share information about for our listeners.
So that you mean that are outside of the research realm, that might be like clinical databases?
Yeah, absolutely, yes.
So here's what I would say. I am going to guess that almost every major hospital and academic health care system has in their packs some pretty valuable traumatic brain images already. And that, because that is, I think, I'm going to go out on a limb here. I'm going to guess that brain imaging is one of the, like for those little babies with HIE, I'm going to guess that brain imaging is almost always done. And so traumatic brain injury is a big umbrella term, like HIE. There's a lot of different reasons why people have it. Car accidents, football, getting shot in the head, huge variation in what that means. And so we have come a long way in developing imaging sequences that are very useful for evaluating many of those different sources of brain trauma. And so there are like susceptibility weighted imaging, for example, comes to mind, which is, was not a very big sequence, when I started this baby brains work, but is now, I think, fairly bread and butter on most acquisitions that have that as the clinical question. I would venture to guess, if I were, I'm kind of risk averse, but I think I would happily put money on a bet that if you had colleagues at various big medical centers who had neuroradiology friends they could talk to and found out were they doing these sequences for those patients, that you could put together a team of people and a team of sites and find this data and work with it. And even though on the research side, we harmonize so explicitly for every little detail of the acquisition, there is a layer of harmonization that is happening across all scanners everywhere, because there's Siemens, there's GE, there's Philips, there's only a few flavors, and each of them only have a few kinds, and each of them only have a few different software versions, and each of them, and if they don't have like our research licensing and special agreements with the manufacturers, they're using the bread and butter sequences that are everywhere, and those tend to be quite good. And so you probably could find some really awesome opportunity there. Call me.
That's great. I wrote down this question before you kind of got to it there at the end about sort of looking at sort of clinical syndrome atlases and comparing sort of patients that you might have against clinical atlases to help clinicians make inferences about where to look. And it looks like, and I wrote this down thinking, wouldn't it be cool to have a tool that does that? And then you went and showed one. So that's fantastic. But I wanted to follow up with that, is that what role is there for data scientists to help develop tools like that? What are some low-hanging things that could get people excited about making those kind of tools?
I don't know what makes this specific to a data scientist. I mean, yes, we should all be doing this. Like, come. So I suppose at the data scientist is a new term for me, because everybody I know I consider a data scientist. So when you ask me that question in that way, can you give me a little bit more guardrails about what kind of an answer you want? Because yes, if you have access to data with, a cohort with some abnormalities that are quantitative that you can find, then there are tools to start working with that. So what are you asking me?
Certainly. Well, I usually like to think of data science as much more broad than machine learning, for example. To say that somebody who does machine learning is a data scientist is sort of, it's neither here nor there, really, because of course they do. But on the other hand, if you wanted to, say, apply machine learning techniques and develop tools around that, what else might they need to know in order to get started doing that?
So So if you're working in this particular little niche domain kind of thing, and you wanted to build those bridges, things that help a lot are being facile with working in the field of clinical informatics. So being able to know and understand what are the warts and whistles in working with EHR data. So missingness and sparsity, and like when absence of information doesn't mean that something's not there. I mean, it's a whole mindset to know how to do data science on electronic healthcare record data. So and a lot of this work is best served when you have a combination of the medical imaging and the detailed clinical information. So for example, with the HIE work, while none of that data that we needed for doing our outcome measures or to do some of the next layers of work that we'll do where we look at factors that influence outcome or more details of care decisions, all of that was gathered by going through the clinical records and pulling it out, mining it out. You wouldn't be able to get anywhere near as much meaning out of those atlases, the atlas application to those cases that have problems without knowing those clinical contexts. The clinical context is really, really important for interpreting the results. And so data scientists who can have enough biomedical knowledge to comfortably, maneuver around in that space and interpret and understand what's there, or at least to be able to ask all the good questions so that they have their domain expert to support them in that way, is really important. I think on the medical imaging physics side, understanding how variations, you have to know which data you can merge and which you can't. And so, for example, with the tool I was showing with Eugenio, where he was doing that post-processing for being able to take the thicker clinical slices and then turn them into isotropic voxels that would run through FreeSurfer, you have to know which, things like you can't use a post-contrast scan because contrast introduces a change in the contrast properties of all the tissue. And so the algorithm is completely different than in pre-contrast scans. You have to understand that the way that the FreeSurfer works on that data, anything that it would output in terms of like a cortical thickness measure is very suspect. It's not really trustworthy the way, say, a hippocampal volume could be. And there's an interaction between knowing the MR physics and how the image is generated and made and the properties of the scan, and then how those algorithms run. And so it's really good to do this in teams because it takes a lot of different people to know all the different parts.
I think that's an important part, is that all of this, and this is one of the reasons I enjoy this, is because it is a team sport, and it really does take a collection of people with different and interlocking skills to be able to deal with the type of data, the amount of data, and all the nuances you just described. So absolutely. Well, Dr. Golub, let me thank you so much for joining us today and sharing your amazing work. I really appreciate it. Thank you once again. And to all of our people who've been listening in today, thank you so much. Everyone enjoy your weekend, and we'll see you next week. Thank you again, Randy.
My pleasure.